Hello, my name is Allison Cusick and I am the co-founder of the FjordPhyto Citizen Science Project. I am also a graduate student in the PhD program at Scripps Institution of Oceanography with Dr. Maria Vernay. FjordPhyto is a citizen science project that takes place in the polar regions on the Antarctic Peninsula with tour vessels and in Svalbard through Hearts in the Ice with Sunniva and Hilde. For the past 2 winters now in the Arctic, Sunniva and Hilde had a unique opportunity to winter over in a hut, B.17, which is located on a fjord in Svalbard. I'm interested in understanding how phytoplankton, or single-celled microscopic algae, respond to melting glaciers. So phyto just means using light for energy or a plant, and plankton means it's a drifter in the ocean, unable to swim against currents, and there's a huge diversity of phytoplankton. Each species of phytoplankton actually contributes, plays a different role in the ecosystem, and so it's important to understand who is there and at what times of the season. So being able to look through the winter at the types of species of phytoplankton that exist is something that is really helpful for understanding how these winter communities might get set up to be ready for that spring when the sunlight returns and the sea ice begins to melt. In the past, researchers have thought that the polar night and winters are times where there's not much activity, where phytoplankton, which are algae that use sunlight to make energy, might not be active. There wouldn't be much photosynthesis going on. And recently, studies have been published that show that there actually is activity through the polar night. Another study recently found that these microalgae or phytoplankton do not shut down their machinery, which would capture light. Instead, they stay ready to receive that light as soon as the polar night ends and the sunlight returns. Now, researchers haven't been able to access places like these to look through the winter, and so when the opportunity for Sunniva and Hilde came along to be collecting samples on a near weekly basis from the fjord, this was something that was of great interest, and that they could help to collect samples that we're able to look at in the lab to see which species of phytoplankton are present. At the end of the first polar winter, Sunniva and Hilde were able to send some samples to me in California, and I was able to take those samples into the lab. And I have a little bit of video to show you guys what that process looks like. Here I am at the Scripps Institution of Oceanography, as you can see. University of California, San Diego. It's a nice sunny day, a little windy, and we're going to go into the lab to see what happens to these guys. This is one winter worth every week through the darkness of the polar night. Phytoplankton from B.17. Now, these samples will get processed by myself, and I'll extract the DNA or genetic information from the sample, and then this will be able to get sequenced so that I can tell what species are present. And I can see things like how the communities are changing through the polar night. This has been a huge effort by Sunniva and Hilde. It's been awesome to get samples like this from this part of the world during a part of time where research data is limited.
We are so thrilled Sunniva and Hilde are bringing awareness to the poles and have partnered as citizen scientists with FjordPhyto. The polar regions are dynamic and changing fast. These changes affect the biology of the ecosystem, starting with food source phytoplankton.
I hope you're all enjoying the Citizen Science virtual conference this year, and I hope you're all enjoying our special feature with Hearts in the Ice. Thank you.